Today I'll be talking about digital clinical measures in Rett syndrome, and in particular we're going to be talking about a novel measure that we're working on with Rett Syndrome Research Trust, ECG Derived Respiration. And just to introduce you to VivoSense, we provide an end-to-end solution for digital biomarkers, and as Theodore mentioned, from wearable sensors. So our focus is wearable sensors. We rely on you guys because you're the experts in Rett syndrome. So we have to have lots of conversations with KOLs in order to understand this better. But let me jump right in. There we go. All right. So first, I'm just going to talk about why digital clinical measures in Rett syndrome. And when I talk about digital clinical measures, I mean things that can be measured with a wearable sensor. We've seen evidence here of some subjective measures, some video measures, which are also digital. But we're talking about where we can be as passive as possible, where we're measuring things without any intervention. We put on a device and we see what we can measure. So some of the reasons to do this are, well, novel endpoints. And we've already seen novel endpoints presented, but novel endpoints from wearable sensors. First of all, we can measure things that haven't been previously measured, such as apneas on a continuous basis. And these may be more sensitive to treatment effect. And now we can look at real world measures as well, which may better represent meaningful aspects of health to the patients and the caregivers. And again, the apnea example is a good example, the breath hold example. There is also potential for objective endpoints to replace some of the subjective patient report or caregiver report that we typically collect, and also to enhance that data and hopefully provide more contextually rich endpoints or contextually specific endpoints. And then we have the continuous data collection as the last piece, where we can expand recruitment because we don't need the patient coming into the site. So we can collect data at home and we can connect it continuously for long periods of time and identify a timing of events, treatment effects, and transient events such as breath holds. So I'm going to start sort of at the beginning where this type of measurement has already been used in Rett syndrome, and polysomnography, which is a sleep measurement, is the gold standard for sleep measurement and has been used to identify apneas, hypopneas, change-strokes breathing, and more. And in order to do that, they actually use respiratory plethysmography, or these two bands. One is around the chest and one around the abdomen, and they're measuring the expansion and contraction of the chest and the abdomen. And by doing that, we're able to identify apneas, hypopnea, tachypnea, and change-strokes breathing just from these respiratory bands alone. And these can be done during awake as well. It needs data cleaning and more, but it can be done and has been done in Rett syndrome clinical trials, both as exploratory and secondary endpoints. So that's the beginning. And now what we want to, let me show you this first. This is a screenshot of some respiratory plethysmography data in a Rett syndrome patient. And what is on the top chart there is the tidal volume with the minimum and maximum events identified, minimum maximum of the respiratory pattern identified. The tidal volume is a combination of the abdominal and respiratory and rib cage bands, which you can see in the second chart, abdominal band in green and rib cage in blue. And then below that, we have the respiratory rate measured, and that's displayed in red, and the TT, which is the total time for each breath, displayed in green at the bottom. And we can quite easily observe the apneas and hypopneas in these data, and using algorithms and some machine learning, we are able to identify these with the software from these respiratory bands and respiratory plethysmography. And this has been done, as I mentioned, in clinical trials in the past. However, what we're looking at here is a novel method to measure this abnormal breathing or these breath holds and tachypneas. And novel method we're looking at is using an ECG patch. So an ECG patch, again, is a wearable sensor, typically the size of a Band-Aid, and you just place it on the chest. And instead of using 2 respiratory bands, you can use this one sensor. The idea behind this is to identify respiratory abnormalities from these ECG signals. We look at R wave to R wave interval timing in order to do this. And from that, we can identify apneas and respiratory rate. So at least that's the goal. And
what's an R wave?
R wave is in the ECG trace, you have a QRST peak, and the R wave is the peak of your ECG beat. And I'm sorry, it's slipping, it's escaping my mind what part of the heartbeat that is. But it's
don't worry. I am, my cardiology is freshman year of physiology. So essentially, it's a distance between what and what.
Yeah, it's a difference between each heartbeat. Basically.
Got it. Thank you.
Okay, so the R wave is the highest contraction of the heart.
Thank you.
And that's what you're looking at, and you're looking at the timing between each heartbeat.
Thank you.
Okay, and we'll display some images in a moment, so you'll be able to get that. So the one thing about this is, because again we're using a wearable sensor, just like with respiratory plethysmography, we would need to look at artifact management, and we need to understand, we need to interpolate additional beats that are being identified, because you're using one ECG patch and you might get conflicting beats. And also because of activity and the fact that this is being done during wake, while the girls may be moving or clasping their hands or doing something else, there's motion artifact and sensor artifact that needs to be accounted for as part of this. And finally, the other step we'd need to take into account is patient's age and respiratory rate, expected respiratory rates. Younger patients have a higher normal respiratory rate, and then Rett syndrome patients on their own have higher respiratory rates overall as well. So these all need to be taken into account. But the first place you need to start is with some sort of validation. Can we actually measure respiration rates from EDR, from ECG derived respiration? And what we did is we took a little study with a capnometer, capnograph, and MC10 ECG patches. MC10, I haven't got any images here, but it's basically a Band-Aid sized patch that can be placed on the chest. So we looked at this, we looked at respiratory rates in different intervals, 30 seconds, 60 seconds, 5 minutes, and then the entire measurement period of the capnometer, which was around 30 to 35 minutes in each recording. And this was done in order to try and do some validation. So we can see a comparison of our ECG derived respiration to the capnometer, and looking at the different trends in 30 seconds, 60 seconds, 5 minutes, and total duration. And we can see quite a close match between the two. The mean respiratory rate in breaths per minute, 19.7 for a 30-second trend versus 21.4 for the capnometer. So we can see actually a reasonably good match when measuring respiratory rate. And the reason we're looking at respiratory rate is we want to try and ensure that we can identify apneas and higher respiratory rates to identify abnormal breathing. So we see a good match there. And taking you through specific measurements here. This, by the way, was a healthy population. And so we asked them to do a 6-minute walk test and then a 2-minute walk test. And we wanted to monitor how close it was. In blue, we can see the capnometer median, and in red, we see the EDR or ECG derived respiration. And we see quite a good match between them. There's a little bit more noise on the EDR, as is expected, because it's a wearable sensor patch, where the capnometer, you're actually wearing a mask and you're breathing. And so we find a good match. This is a 30-second trend. We looked at the 60-second trend. Once again, good match. And even during the 6-minute walk test and the 2-minute walk test, we see increases in respiratory rate that we expect. So that's good. And then we looked at a 5-minute trend. And once again, good match between the two devices. And there's a total duration, and we can just see a bigger standard deviation, more range on the EDR, once again in the red compared to the capnometer, but once again, a good match between them. So with this in hand, we wanted to pivot towards Rett syndrome and abnormal breathing and see what we could do. So we'll talk first about the benefits over respiratory plethysmography. I've mentioned this already a little bit, but it's less intrusive. When you do respiratory plethysmography, you've got 2 bands, they have to be relatively tight around the chest and abdomen. Typically, it's a vest that's being worn. Hexoskin makes one. The vest is quite tight to wear. Sometimes the girls don't like it so much. An ECG patch is much easier. You just put it on, and it can stay there for up to 14 days. Some ECG patches can last up to 14 days. We already know from our previous study that it measures respiratory rate with similar accuracy to capnometers. We know that respiratory plethysmography has also been compared to capnometers and has a good correlation there as well. So we see a potential for detection of apneas or breath holding during wakes, and potential to detect tachypnea or increased respiratory rate during wake as well. So we're working with Rett Syndrome Research Trust on the Emerald study, where they're collecting ECG from MC10 biostamps on up to 20 Rett syndrome patients. We have a in-clinic measure of respiration with respiratory plethysmography simultaneously that we can use as ground truth in these data. And our intent is to compare the respiratory plethysmography to the ECG derived respiration, and looking specifically at apneas in the first part. And so we'll show some data here. Top chart in green, we have the tidal volume from respiratory plethysmography. And once again, we can see some apneas occurring, some breath holds occurring. In the red below that, we have the heart rate derived from the ECG channel. And in the blue below that, EDR is the ECG derived respiration. So the EDR is like the tidal volume channel, but derived from ECG. And then you can see the peaks being identified in yellow and red below them. So we're identifying breaths in this blue chart over here. And at the bottom, we can see the TI, which is time of inspiration, for each breath as it's reported. And what we'll note is that for the most part, the time of inspiration of the ECG derived respiration matches very well with the time of inspiration being increased for the tidal volume being measured by respiratory plethysmography. And these TI, EDR are events that were identified with algorithms and some machine learning to identify apnea or breath holds on inspiration from the EDR, and we're trying to match that up to the respiratory plethysmography. So we see once again that this is looking very promising. We only have very few files at the moment. We have 3 that we're working with, 3 subjects that we're working with. So we're trying to gather more of the remaining 20 subjects that we're working with here. But so far, good outcome. And here, just taking a closer look, this is in the yellow, and I'm sorry, you can't see the identification below, but it's an apnea identified from the respiratory plethysmography, and we can see that it's overlaying almost exactly with what we're identifying from the EDR. So once again, good match and promising results for us to identify apneas from ECG patches. So to conclude and to just sum everything up, because I think that digital outcome measure there's an opportunity to use digital outcome measures more in Rett syndrome. I know we're having many conversations with sponsors and obviously with the support of the Rett Syndrome Research Trust. We appreciate that. And there's an opportunity to really provide some novel outcome measures from wearable sensors. EDR is one of them. I know Dr. Downs has done a lot of work also in behavioral activity, and we're excited about that because you can use wrist-worn actigraphy to do measures such as that and look at, instead of a 6-minute walk test, we can look at some form of activity during the day that can give us some sign of the girl's behavior or activity behavior. And we can try to address more meaningful aspects of health for the patient and for the caregiver. And this will lead to more primary endpoints in clinical trials for our sponsors. And the last point, of course, is with wearable sensors, we can do continuous monitoring for days, weeks, months at a time, with the right sensor, with the fit for purpose sensor, and the right events being, the right outcome measures being measured. And that's the end of my presentation. So thank you.